right, guys, so for this week, we're going to be studying cells. So this is going to be a long chapter, so find your favorite snack or find a good pillow to snuggle with because this is going to be a long lecture. All right, so let's go ahead and get started. All right, so for cells, so if you should remember cells on the hierarchy of after atom molecules, and then you had organelles, and then we had cells. So we're starting to move up in the hierarchy. So cells are essentially the basic living thing that we have, the smallest living thing that we have. And in order to see cells, we have to use what's called a microscope. Microscope stands for micro. That prefix means things that are small. And so we have to have something that's able to see at that magnification. So there's a few different types of microscopes. So you have light microscope, transmission microscope, or electron microscope, and then scanning electron. So light microscope, essentially those specimens that we look at have to be so thin that light can pass through them. And that is what's going to allow us to be able to see those pictures or images in the microscope. Transmission electron microscope is going to essentially pass electrons through it. That's going back to chapter 2 with chemistry. It's going to pass electrons through it to kind of give us a little bit more in-depth picture, and then scanning electron allows us just to see the top surface a little bit more intensely. So with light microscopes, which is typically what we use in lab, they only produce a two-dimensional image by passing visible light through the specimen, like I said earlier. An electron microscope just uses a beam of electrons to illuminate a specimen. So basically, it kind of it shines essentially a bright light on it. It just kind of gives us an idea of what it looks like on the surface. And in transmission, it actually shoots direct electron beams through a thin slice of the specimen. And it can even get to the point to where we can magnify it by 50,000. So that's incredibly small, and we can see it in a very big picture. So, I mean, we have to get a lot of magnification to be able to see it. Then we have scanning electron microscope. Essentially, there's a beam that's moved across the surface of it, and which only gets to about 3,000 magnification. So not too much as transmission, but it is we're still able to see a little bit more details than a light microscope could. So starting off with cells, the human body has around about 50 to 100 trillion cells. If we had $1 for every cell that we had, we'd be very, very rich. But unfortunately, we do not. But it's crazy to think how our body has that many cells. So just to kind of give you an idea how small cells actually are, we can fit about 5 million cells, 5 million blood cells or red blood cells. They can fit on my pen right here. That is how tiny cells are. I mean, that's kind of like the needle or the point of a needle at the end of my pen here. I can fit 5 million red blood cells that are in your blood on the tip of this pen. That's pretty cool to think about. Some cells have different shapes. Remember, shapes determine function, or structure determines function. So some can be cubed, column, cylindrical, disc-shaped, or irregular shaped. So understand that the way the cell is structured, or the shape of the cell, determines its function. So cells that are in our bones are not going to look like cells that are in our eyes. Cells of our skin aren't going to necessarily look like the cells that make up our teeth or that make up our bones. So each cell is going to look different just because of where it's placed in the body and what's its function, but they all have the same components. whether that be a nucleus, a mitochondria, it all has the same components. So here it gives us a good picture, a representation of how cells look. So this is kind of giving us a size chart. So in comparison to a human, we can see some muscle cells if you put them really closely compact together. We can see an ostrich egg. How we can barely see a human oocyte or a egg. So everything here in the blue is actually showing us what we can see with our naked eye. However, once we start getting a little bit more microscopic, like looking at cells, bacteria, red blood cells, or mitochondria, it's going to need to have a light microscope, which is something that we use in class, to see things that are a lot more small than that, such as viruses and ribosomes and proteins and amino acids. We need an electron microscope that is a lot more sturdier, but also that can see at that high resolution and magnitude or magnification. So here are some different red blood cells, well, actually just cells in general. So we have irregular shape like our nerve cells. We have biconcave like our red blood cells. That kind of looks like almost like a donut without the donut hole. We have some cube-shaped cells that we're going to find kind of in our kidneys. Some column shape, which we can find potentially in our throat, but more in our gastrointestinal tract. We have some spherical cartilage cells, which you're going to tend to find in around our bones, especially where we have two bones articulate with each other. And then we can find some cylindrical that are going to be in our skeletal muscles. So here's the cell, and here we have a lot of different components that make up that cell. So here, which is usually very identifiable, which is going to be our nucleus. Usually the nucleus is pretty big and it takes up a good portion of the cell, at least 25% of the cell. If you look here, it does take up a good portion of the cell. So the nucleus is what's going to house our DNA. And do understand that DNA doesn't typically leave the nucleus. It's always going to stay housed in there. This little blue portion here that's around the outside is going to be called the nuclear envelope. Just like you have an envelope in a letter, that's going to be the outside of it with the letter on the inside. The envelope is the same here. It's the portion that's on the outside of it. And here, which it doesn't have it labeled, but these little parts that are right here, these are actual little ports, also called nuclear pores. And so that allows for things to come in, small things to come into the nucleus and be able to go back out. So membrane-bound organelles. So membrane just means that there's a portion on the outside of, well, actually, it just means that there's a portion on the outside and there's a portion on the inside. So it means that there's something that's protecting that organelle. In this case, we have the rough endoplasmic reticulum, which is always going to be found around the nucleus. And it's called rough, not because that it is rough, but it appears that way because all those little ribosomes that you actually see, that's all attached to the rough ER. Those are going to make it appear rough, but it's not necessarily rough. It's just it has a bunch of ribosomes attached to it. This green portion you have here is referred to as the Golgi apparatus. So that is actually what we call the mail or the post office of the cell. So the Golgi apparatus is responsible for packaging and shipping proteins and things that the cell needs to outside the cell or bringing it into the cell. So it kind of works just like our FedEx or UPS. That's actually kind of how it works. It actually just packages up things to be shipped out the cell. The mitochondria, which most people know as the powerhouse of the cell, this is a representation of how mitochondria actually kind of look. Mitochondria are for producing ATP, which is our energy molecule. So they are very at work, and sometimes in some cases in some cells you may have 50 to 40 or 40 to 50 mitochondria per cell, depends on where it's at. So like muscle cells will have a ton of mitochondria, just because they are constantly needing energy. Bone cells may not have very many mitochondria at all. So it depends on how well that or where that cell is actually located. Let's see here. The plasma membrane, which is this membrane that goes all the way around the cell. The plasma membrane is essentially like the gate or the fence that protects the inside of the cell. It allows things in and it keeps things out. So you want a plasma membrane. That membrane only allows certain things to come in or go out of the cell so that the cell doesn't lose things that it needs or keep things that it actually does need or prevent things from entering into the cell. So essentially it's like our skin, it's our first line of defense. So it has to have something to protect the entire cell, and that's referred to as the plasma membrane. Some cells have microvilli, which microvilli are for absorbing materials. We're going to find things like this in our gut. That's when our food gets to our gut. These microvilli, which a lot of cells have, will essentially absorb our food. So microvilli are for absorption. Cilia. Some cells will have cilia, which cilia are essentially hair-like projections. So they kind of underneath the microscope, they kind of look like little bitty hairs. We're going to find things like this kind of actually in our trachea or in our windpipe. So when you're coughing up mucus, these cilia here are actually coughing up. They're actually moving in wave-like patterns to actually move up the mucus out of your throat so that you can cough it up. Also, you can find cilia in the fallopian tubes. That's what's actually going to help push the egg to the uterus. So these are actually kind of cool little features that cells do have. But cilia are not found in every cell. They're only found in certain places of the body. Just like flagella, flagella is actually used to move the cell. So it actually kind of acts like a little tail or a propeller. The only cells that usually have flagella are going to be sperm cells because they actually have to move, or as we say, swim. But not too many cells are going to have flagella. Vesicles, which actually, if you look here, it actually looks the exact same as these around the Golgi apparatus, and that is because it is. Vesicles break off the Golgi apparatus, and they can actually carry things or proteins to the plasma membrane to release it from the cell. So vesicles are really just the Golgi, but they just have been pinched off to make a little separate little pod that actually is going to travel and go to the border or the edge and actually release their items or capture their items. The cytosol, which is what this clear fluid is that you see that all these organelles are floating in. So it almost looks like the cell is floating in like a pool of water, but it's not a pool of water, I promise. It's really what's called the cytosol. So it's the intracellular fluid. So intra means inside. So all this is it's floating in a fluid. Now this fluid is kind of viscous, kind of like almost like a jello kind of mixture. So it kind of just sits, all these organelles kind of float in the fluid. These large components here are referred to as lysosomes, which lysosomes are responsible for breaking down materials. So lysosomes also have the name of being called the garbage or the garbage disposals of the cell. They're going to get rid of a lot of unwanted things that the cell doesn't need, or if they're disposing of things, or if organelles are dying, or parts of the cell are dying or getting replaced. The lysosome is going to break those components down and then recycle them whether that be for making new nucleic acids, making new proteins. It's going to essentially break it down and recycle it. When you cry, if you get dirt in your eye and you start to cry, your tears actually contain lysosomes. Lysosomes are going to essentially break apart the bacteria. If your body can use some of those components, it will. If not, it will dispose of them. So that's actually your lysosomes. There's lysosomes in your tears, especially when you get something in your eye. Peroxisomes, it actually sounds like what actually makes what it sounds like. So peroxisomes actually make peroxide. So what's actually happened there is going to help. It kind of does the same job as lysosomes, but it can actually break down other components. It especially has a role in apoptosis, which is cell death. So it actually helps break down a lot of things as well. So you have bound ribosomes, which you're usually going to find attached. That's what bound means. It's attached to the actual rough ER. And then you have some free ribosomes, which are just free floating, which are just floating in the cytosol. So either they can be attached or they can be just floating. Then you have what's called a cytoskeleton. So with a cytoskeleton, just like we have a skeleton, which is for our structure and posture of our bodies, each cell has a cytoskeleton, which is going to help keep the shape or the form and the structure of that cell. So it doesn't look like our bones, of course, but there is proteins that are shaped in a certain fashion that's going to help that cell stay, if it's a circle cell or it's a cube cell, to stay in that shape. The smooth ER, which is called smooth because it looks smooth, but it's smooth because it doesn't have any rough or ribosomes actually attached to it. So it gets the name smooth ER. And that's the reason why, because it just doesn't have any ribosomes attached. Smooth ER is really responsible for producing lipids, breaking down lipids, and also breaking down alcohols and steroids. So anytime you go drinking on a Friday night, those smooth ERs are working overtime to actually break down the alcohols that you drink or consume and things of that nature. And I believe that is most of everything for the cell, pretty much. As for identifying some of the key structures, which it goes into detail underneath the picture as well. Now, for the plasma membrane, which it has a very, very particular component and structure. Do you understand that all the plasma membrane isn't just a membrane? It's actually composed of phospholipids, and those phospholipids form a bilayer. So in the previous chapters, we know that the head of the phospholipid is actually hydrophilic, which means it likes to interact with water. The inside of the layer here, which this is the very inside here, where the tails are, this is hydrophobic. Remember, those tails are the lipid components. So lipids, or which are oils, don't like water. They're hydrophobic. So this inside environment here, which where these tails are floating against each other, that part is actually very hydrophobic, which there's no water that passes through there. And so here you can see what's called proteins, or these channels here. So this one particularly is called the integral protein. Integral is essentially going to go from the outside of the cell to the inside of the cell, so that things that are too big or polar can actually come through and go from the outside to the inside. So it has to have like a little pathway or tunnel that it can actually kind of skip this and go across. Remember that the phospholipid bilayer is polar, well, it's non-polar. So some things are too big to get in between these phospholipids. So they have to have a channel or a secret passageway to get from the outside to the inside. So the things that you see here that are linked to each other, these are what's called glycoproteins. Glyco meaning sweet or glucose. And then you have some that are, you have the carbohydrate. So these are essentially like antennas for the cells. And you might be asking, why do you need an antenna on a cell? Well, they're for essentially being able to recognize other cells or for the immune system to be able to recognize them. So glycoproteins are essentially like cell markers that are going to identify that cell or that cell belongs to the body. So these are very, very pertinent when it comes to your immune system. If you lose these, your immune system, or if your immune system loses the compatibility to recognize these structures here, you're going to have what's called an autoimmune disease, where essentially your immune system starts killing your own cells. So cholesterol, cholesterol is a lipid. However, we need that. So if you look in the phospholipid bilayer here, you see that there is cholesterol, and we need cholesterol. Cholesterol helps keep our plasma membrane fluid. So even though we're seeing this as a picture, these phospholipids are actually moving. So it's frozen here like in a wave pattern, but they're actually moving up and down. So it's very, very flexible. And so this cholesterol is actually good and keeps it flexible, especially in the wintertime. We need to have cholesterol. So it's like fish when they're super low in the water and let's say the lake freezes over. What keeps them alive is them having cholesterol in their cells. By having cholesterol in their cells, it keeps their cells fluid, allows for them to essentially keep living. If not, their cells would freeze to death. So think about this, you can't really freeze oil, right? So by there being cholesterol in their plasma membranes, it keeps it fluid, it keeps things flowing properly. So every time we hear cholesterol, we always think that it's a bad thing. It's actually a really good thing that we have cholesterol. We need cholesterol. However, there's a good cholesterol and a bad cholesterol. And sometimes bad cholesterol kind of gives the good cholesterol a bad name. But, so we have the integral proteins, which is going to allow things from the outside to the inside to pass relatively quickly. Sometimes we have peripheral proteins, which are in the peripheral, which means it's kind of to the side of that protein channel, which is going to act like a messenger, which we'll see further along in this chapter. So again, these are just little receptors and antennas that are going to allow for connection between other cells or recognition of other cells, and especially the immune system. So here are some different types of transport proteins. So we have a channel, which we saw earlier, which allows things to pass through. We have what's called a carrier protein, which is going to essentially chaperone things through. Sometimes you'll have a pump, which is going to essentially do like what you think it would. It's actually going to push things against its concentration gradient. You have what's called a cell surface receptor, and it binds to what's called a ligand, which is just simply a chemical or a chemical structure that's going to bind to the receptor and cause a response. We have the identity marker, which we saw earlier. And then we also have what's called an enzyme, which we saw in the previous chapters. So when it comes to the membrane, there's some things that can go across, some things that cannot. But the things that do get to go across, it's called membrane transport. But there's different ways that this actually takes place. So some things are passive, which means it does not require energy. Some things are active that requires energy. So anytime you hear of someone being active, let's say that they're running or working out, that requires energy for them to do that. So active means energy. Passive is the opposite. Passive, you do not require energy. So this is like me rolling a ball down a hill. You don't need energy, it just rolls down the hill. But if I wanted to take that ball up a hill, that requires energy. So if I rolled it down a hill, that would be passive energy. Just letting it roll by itself. It doesn't need any energy, it doesn't need me to really push it or anything or carry it. It can just roll down the hill. Active processes or active transport, that's going to require energy. So that's like me taking a ball at the bottom of the hill and walking it up to the top. That is active energy. So let's focus on passive. So with passive, there's a few different ones. You have what's called diffusion or osmosis. Diffusion is just movement of solutes, and osmosis is movement of water across a semi-permeable membrane. So with diffusion, if I was to take some cologne or perfume and spray it in the corner of a classroom, by the end of the class, you'd be able to smell it all across the classroom. That's because it went from a high concentration to a lower concentration. So that's just like me rolling a ball from the top of the hill to the bottom of the hill. It's going to go from high to low without any energy. So again, if I sprayed perfume in a corner of the room, it's going to be really concentrated in that corner of the room because that's where I sprayed it. But eventually, all those molecules will spread out, and then you'll be able to smell it everywhere in the classroom. So that would be a example of diffusion. Osmosis, on the other hand, that is like me taking food coloring in a glass of water, and I just put one drop of food coloring in the water. And at first, it's going to drop to the water, and then eventually, when you come back to it, it's going to have went from that one drop in the middle of the glass, and the whole glass of water will be that color. So that would be osmosis. So it's literally doing the exact same thing, just one is essentially kind of in the air or anything else besides water, and then if it's in water, it's just dealing with osmosis. Now you have what's called, in diffusion, you have what's called simple diffusion or facilitated diffusion. Simple just means that there was no protein required for movement, meaning nothing had to be used to make something diffuse, essentially. Facilitated means that it had to be helped. There was some type of protein or transport protein that was going to have to be utilized in order to make things move. And then you can get into channel mediated and carrier mediated. So channel, basically what we saw earlier in the plasma membrane, where you have that channel going through from the outside to the inside, or you can have carrier mediated, which we saw in the last page up there as well. With active processing, so you can have what's called active transport, where ions are pushed against their concentration gradient. So this is saying if we had let's say we'll use the ball example again. Let's say we had a bunch of balls at the bottom of a hill. We now got to carry them up a hill, even though they want to be at the bottom of the hill. So that would be a form of active transport. So that we had to gather all those balls and go up to the top of the hill. That would be active transporting. That means we're taking against its concentration gradient. You're going against what it wants to do. With vesicular transport, which involves a vesicle, which is what we saw pinching off from the Golgi apparatus. So you can have what's called exocytosis or endocytosis. Endo means coming into the cell. Anytime you see EX, which is X like exit, it's leaving the cell. And then for another form of endocytosis can be what's called phagocytosis, which essentially it looks like a little Pac-Man that's going around and actually eating up either debris or bacteria. Or you can have what's called penocytosis, which is referring to it's doing the same thing just in liquid. So we tend to separate these in between phago and peno, which is just phago is just like actual materials versus peno is just water. All right, so let's give you some examples of diffusion. So with diffusion, or actually osmosis, but osmosis is just a diffusion of water. So if we were to drop in some food coloring, we'd see it actually spread out, and that would essentially be diffusion would be the spreading, but once it spreads out completely to where you can't see the difference, it's actually going to be at what's called equilibrium. So it went from a higher concentration, and then it's going to spread out to everywhere where it's a lower concentration to where to the point to where everything's going to be equal. So this would be diffusion essentially, or even osmosis, or a simple diffusion really. With this one, it's going to show how things are going to go across the membrane. Sometimes it takes a while because the membrane's kind of really tightly packed, but some things can kind of be small enough they can actually pass through the membrane. If you look here, look as if this is a single file line of people. Only a few very small things can actually get in between these. So some things can diffuse across the membrane if they're small enough, but if they're too big, they're not going to get across. You're going to need something like this. So here you actually see ions or elements that we saw in chapter 2. They're actually moving across the membrane. So in order to do that, they have to have a secret passageway or a channel that's going to allow for things to actually go and pass through. So here you're seeing things move down, what's called down their concentration. So they have a higher level here and a lower level here. So things will go from high to low. Here we have a higher concentration here and a lower concentration there. So you're going to see things go from higher to low. So think about it like a ball rolling down a hill. You got a ball at the top of the hill is going to roll down to the bottom. Same thing here. Here we have what's called carrier proteins. And so carrier proteins change shape to transport small polar molecules. across the plasma membrane. So here we see glucose is coming in. You have the glucose carrier protein, which is going to allow for glucose, which is a big molecule, because remember, this is a 6-carbon ring molecule, to actually pass through here and get into the cell. And so sometimes with these carrier proteins, that's why you have to have insulin. Insulin is going to bind to these, which is going to allow for glucose to actually come into these carriers so they can come into the cell. So here's a really good representation of how our plasma membranes are actually working. So you see a lot of osmosis, which is the passive movement of water through a semi-permeable membrane. So here we see water actually coming across what's called an aquaporin. So if you think in Spanish what water is, it's aqua. So aquaporin is literally a pore where water can pass through easily. Remember, water is very polar, so it's not going to be able to really go through the plasma membrane very easily. That's why they show you like only one water molecule being able to pass through here. But we have a channel that allows water to pass through a lot easier without having to try to go through the plasma membrane. So here we can see that we have different molecules. We have some calciums, some chlorines, some sodiums, some glucose, and we see that we have higher concentrations on this side and a lower concentration over here. So things are going to go from high to low. So we're going to see things over here. Going to want to get to this side over here. Some things can pass the membrane that are small and non-polar, but if you are big or larger or polar, you're going to have to find a channel to get across, so like a little bridge, because you can't just climb across. So here's just a really good representation of how that works. So proteins, which are really big, they have to have their own channels. This protein really wouldn't be able to fit through here. It's only big enough for what's supposed to be going through there. So if it's an aquaporin, only water can go through there. These will actually have to have a protein channel that's big enough to allow for things to go in, or a vesicle to actually transport those through. So this is just a good representation of what our plasma membranes look like. So each cell, which we have on average about 75 trillion cells, each cell has a membrane. Each cell has to have a membrane that surrounds the cell, that keeps the inside on the inside and the outside on the outside. So we have to have something protecting each cell, and that protection also allows for things to go in and out of the cell. So why is that even important? So if you're going into the medical field, this is very, very, very important. So one thing you should always remember is that water chases salt. I want you to write that down. Water chases salt. So when it comes to water concentration and solute or solute concentration, water will always either chase salt or the solute. So let's say you can use salt or sugar in this case. So water is going to go where there's a higher concentration of salt. So let's take a look at these pictures here. So with this one, in this isotonic solution, it says the interstitial fluid, which is the fluid that's around the cell, is the same as the concentration as the cytosol, which is the fluid on the inside of the cell. So if the concentration on the outside is the same on the inside, there's going to be no net movement. There's no higher concentration, there's no lower concentration. They're both the same. So that's isotonic solution. Now, let's look at the difference between the isotonic and the hypotonic. So hypotonic, if you notice, it's a lot more swollen up. I tend to remember hypotonic by, it reminds me of a hippo, which are really, really big. So I think of hypo when it comes to water swelling. So what's happening in hypotonic there is more salt on the inside of the cell than the outside of the cell. So water is going to chase salt or sugar. So the water on the outside of the cell is going to go on the inside of the cell so that it can make it balanced. And so that cell ends up swelling or getting bigger because there's a lot of water moving into it. So this is like when you let something soak in water and it becomes bigger because it has a lot more water content. It's the same thing. We used to do a gummy bear lab where we just drop water or drop a gummy bear in water, and it will essentially that gummy bear would get bigger and start soaking up that water. So because it had a lot more solutes in it. So water, always remember, water chases salt and sugar. In this next case here, we have a hypertonic solution. And I think of hyper, I think of like a little kid throwing a fit, because that's what it kind of looks like here. Like he's throwing a fit, or he's hyperactive. So if you have more salt on the outside of the cell, the water that's on the inside is going to chase that salt. So that water is going to leave that cell. So remember, water chases salt. So we see there's a lot more salt on the outside of the cell. So we're going to see the water that's on the inside of the cell leave and try to go and find the water. This is important when it comes to like IV bags. IV bags or intravenous bags that you get at the hospital where they have to start an IV, especially like with saline solution. Saline solution has like a 0.9 salt concentration. It's the same concentration of our bloodstream. So that would be this isotonic solution right here. So IV or saline bags that we get at the hospital are actually isotonic solutions. It has the exact same amount of salt that our cells have, and we want that. We want our cell shapes, our red blood cell shapes, to remain the same. So let's take a look what would happen if we put distilled water in a vein or an IV bag. So distilled water has no ions in it whatsoever. The only thing that's in distilled water is H2O. In isotonic, you have some salt. However, in distilled water, there is nothing but water. So if you put distilled water in our veins, there's going to be more salt in our red blood cells than, of course, that's in the fluid or the water that's around our blood cells. So that means that the water or the distilled water is going to flow into our blood cells and actually expand them and potentially bust them open, which means the red blood cells aren't going to be able to work anymore. So that would what would happen if you put distilled water in someone's veins. For hypertonic, so if you've ever been to the ocean, you know that the water is very salty. And they always say if you drink salt, you can't drink salt water because it dehydrates you. Well, you're probably thinking like, well, if you're drinking water, what's the big deal if it's salty? It's water, right? No. So when you drink salt water and that water gets into your veins are going to have a lot more higher concentration of salt now. And your red blood cells, or your cells in general, all the water from your cells are going to want to leave your red blood cells. And so now they are going to be kind of shrunken. And we call that crenation, which is this word that's right here. So all that water from your cells are going to leave. And so essentially they shrink. And remember, structure determines function. This structure here in isotonic, this is what our blood cells are supposed to look like. And that's how they function normally. But if they change shape, they change their function. And sometimes in these two cases, if you change the shape of the red blood cell, they can't carry oxygen. And if they can't carry oxygen, they can't deliver oxygen to your cells, and your cells begin to die. So if you were to put distilled water in someone's veins, or if you were to drink salt water, a lot of salt water, you can potentially dehydrate yourself with the hypertonic solution and possibly kill someone. But if you put distilled water in someone's IV, you could definitely kill someone. So it's very pertinent that you understand toxicity of solutions. And here it's actually giving you actual images of blood cells that have been in isotonic solutions, hypertonic, and hypertonic solutions. Also, when you drink alcohols, the same thing happens. So that's one reason why you wake up with a hangover, is because your cells are dehydrated. So all the alcohol, let's say instead of this being salt, this is alcohol, that pulls all that water out of your cells, and then especially out of your brain cells. And then when you wake up in the morning, you have a big hangover. It's because you are dehydrated. So that's what's actually happening. What causes a hangover is your body is dehydrated. That's why they tell you to drink electrolytes to replace everything, especially because alcohol gets rid of a lot of your electrolytes and a lot of your salts. So you gotta be careful with that sometimes. So in some forms of movement, you have what's called a symport and an antiport. So symport means that it's transporting to this channel can support the movement of two different things. So here you see glucose being moved out or in, and this one's also moving sodium in. So this has the capability or capacity to move two things at once. Or actually, they move two things at once, but in the same direction. Antiport means that it moves one thing one direction and another the opposite. So here we see sodium coming in, but here we can also see protons or hydrogen going out. So it's an antiport, means it's the opposite. So one can come in, one can go out. But symport, two can also come in, but they both just come in. So glucose comes in, and then sodium also comes in. So that would be the difference in antiport and symport. And ports are important because this is how things get inside and outside the cell. So if you need to pump something out, you got to use essentially an antiport or some type of channel. But symports just pull things straight in or straight out. Vesicles, which it gets its name from like the Latin root of vesica, which means bladder, which means it can hold materials. So this is actually pretty cool. So vesicles, to get stuff in or outside the cell, they get to fuse with the membrane. And so the best way I can think about how this actually works to give you a visualization, which here you can see that the vesicle approaches the cell membrane and then attaches to the cell membrane, and then it essentially becomes a cell membrane, and it disposes of what was on the inside of the vesicle to the outside of the cell, which is pretty cool. It just kind of like it bonds with the plasma membrane, and then it just expels its contents on the outside while it becomes part of the membrane. So the way to think about this is like if you have a refrigerator that has those two double doors, and you go up to the refrigerator and you grab onto the door handles, which would be essentially steps 1 and 2. And then 3 is when you actually start opening up the door handles. So now the door is connected to your hand, to your left hand, and you got a door on your right hand, and you're all part of that same door kind of thing. So that's actually kind of what this reminds me of, which is pretty cool of how essentially our cells gets rid of some things. Also, this can work in the reverse order. So it can actually take in materials. So things are there on the outside of the cell. It can essentially make an invagination and essentially work backwards, and it will carry what was on the outside of the cell on the inside of the cell. So this is a pretty cool thing of how our cells are actually able to ship things out and bring things back in. So essentially these vesicles come from the Golgi apparatus, like I said earlier, and they're able to ship things out or bring stuff in, which is pretty cool. So another way that vesicles work that we can actually put into like a medical perspective is hypercholesterolemia, which hyper means above, and cholesterolemia means that there is an elevated, or this is referring to cholesterol in the bloodstream. So emia means in the bloodstream or in the blood, or cholesterol means cholesterol, of course. So this disorder is actually inherited within genes. And so we have what's called low density lipoproteins, or LDLs, which this is referred to as our bad cholesterol. So the purpose of LDLs is essentially it detects when there are lipids in our bloodstream, and then they pull it into our cells. The vesicles pull into our cells and degrades them. So this is essentially what this was showing up here. So we have, so let's say this is the outside of the cell. It's going to pull in the cholesterol, bring it into the cell, and actually start degrading it. However, in this type of disorder, that's a defect that doesn't happen within the cells. Why is that a bad thing? So if you're not able to break down cholesterol fats, you have a potential of allowing those lipids and fats and cholesterol to just kind of stay in your bloodstream, which I have it highlighted down here. It can essentially build up great levels of blood cholesterol. It can cause plaque buildup. It can cause your blood vessels to narrow, which is referred to as atherosclerosis. It can result in blockage of coronary arteries, resulting in a heart attack, coronary heart disease. Essentially, what this big word just means chest pain. And so, in certain cases, especially in your teen years, it can actually cause a heart attack in your teens and potentially death. So these vesicles do a really big job. It's responsible for getting stuff out of the cell and bringing it into the cell. So if you can't get something out, it could build up inside the cell. If you can't get something in, it can build up inside the rest of your body. So vesicles actually have a really big component or job that they do. And so if it's taking something into the cell, again, endo means inside, that's endocytosis. If it's expelling something, it's exocytosis because it's exiting the cell. So be mindful. Here we see what's phagocytosis. So phagocytosis is essentially when it kind of eats it. So when I think of phagocytosis, I think of Miss Pac-Man, and it's going around because they actually do go around like this and actually eat things and debris that's floating around in our bodies that not supposed to be there. So if you look here, when the body detects, or the cell membrane detects that this particle is not supposed to be in the body, that plasma membrane starts to fold around it and makes its own vesicle. And then that vesicle is going to actually start releasing lysozymes and enzymes that's going to start breaking this down, which is referred to as cellular eating. Here we have what's called penocytosis, which is what's referred to as cell drinking. So when it's just dealing with fluid, phagocytosis is dealing with particles, like actual physical particles. Penocytosis is dealing with just liquid. So they do the exact same thing, just one is dealing with something solid and one is dealing with one that's liquid. And so it essentially makes a vesicle, and it's going to start breaking things down in that vesicle. The reason it does this is because either it's trying to prevent it from causing infection, disease. So that's one reason why it's important for those glycoproteins to be present, so that the cells recognize that those are supposed to be with those cells, or that it's part of the human body that it's in, so that it recognizes if it's its own cell or not. So that's one reason why it's very, very important to have those glycoproteins, because if not, your immune system will start attacking its own cells and essentially start killing its own human host, really. And then here you have what's called cell receptor mediated endocytosis. So if something attaches to a receptor on those cells, it automatically tells that plasma membrane, go ahead and start invaginating and start breaking whatever it is down. So this is a way kind of how if we get a virus or some type of bacteria, those bacteria and viruses will bind to our proteins or the receptors on our plasma membranes, and a automatic response from the body or that cell will start to actually break down because it recognizes, oh, this is not my cell, this is something that could potentially harm me, let me kill it. So it's going to start making its own vesicle, and it's going to start breaking it down and killing it so that it doesn't essentially kill you. So resting membrane potential. So this is actually going back to the very beginning of the chapter. So resting membrane potential is essentially the membrane at rest. I know that sounds very intuitive, but that's actually what it is. So here we have a plasma membrane with some different channels. So if you look here, we have some different ions. So we have some potassium and then some sodiums. And we have some different concentrations. We have a concentration of sodium out here and a concentration of potassium on the inside. If you may remember me talking about it in chapter 2, if you have different charges or different ions on each side, it will cause an electrical impulse or electricity to occur. So here we have more sodium on the outside of the cell and more potassium on the inside of the cell. So if they have different concentration gradients, they're going to try to go to the opposite side. And that's what these channels are for, because remember, these are too big to fit through here. So on this side of the cell membrane, you can see all these positives. The outside of the cell is a lot more positive. On this side of the cell, or this side of the membrane, it's a lot more negative. So this is what is referring to of membrane potential, meaning that there is a electrical difference across the membrane when the cell is at rest. So just remember that there is more K or potassium on the inside of the cell, and there's going to always usually be more sodium on the outside of the cell. So that gives the membrane a charge. And so when we get ready to essentially make electricity, and we see ions starting to flow, that's going to create electrical charge or impulse. So when it comes to cell communication, there's a few different things that can actually happen. So we can have what's called initiation or binding. So binding to a receptor is going to initiate a mechanism that's going to control growth, reproduction of cellular process of individual cells. So we have what's called channel-linked receptors, which permit ion passage either into or out of the cell. So to break that down into simpler terms, essentially where we have ions like potassium, calcium, chlorine that are going to bind, or something's going to bind to that cell, which is going to act like a key to like a gate, which is going to open it up and allow for ions to flow through it. That is a channel-linked receptor. We'll talk about that, especially when we get to muscles. So we'll have, or also when it comes to even insulin and glucose. Insulin is a channel-linked receptor. So literally insulin comes in, unlocks the gate, and then glucose is allowed to come through. But if you don't have insulin, glucose does get to go into the cell. So it's going to essentially linger outside the cell, which means that the blood gets sticky and it can cause other issues with the bloodstream. And so they end up having diabetes. So that is just an example of a channel-linked receptor. Enzymatic receptors, so they function as protein kinase enzymes. So they directly phosphorylate, which means it just adds a phosphorus or can take away a phosphorus enzyme in the cell. So understand that enzymes can either be turned on or off by adding a phosphorus or taking away a phosphorus, like we mentioned last chapter. So G-coupled proteins, they serve essentially as an intermediate molecule. And let me actually give you a representation of that. So here in A down here, we have channel-linked receptors, which means you have to have a ligand bind, which is going to unlock the gate. So we see here, it hasn't bound yet, so the gate is locked. Once it binds to the channel, it's going to open up the gate to allow ions to pass through. So that is an example of channel-linked receptors. Enzymes, we have a ligand that's going to bind, which is going to allow for the enzyme to be turned on or off by the phosphate ion. But down here in G protein, which I think are very, very interesting. So in the first part here, you see the ligand coming in and binding to the receptor. And then it's going to, that G protein that's connected down here on the other side is going to go over to the channel and unlock it. So this may look confusing, but let's think about it like this. So let's say you have a mailman and a butler. So the mailman rings the doorbell, which is the ligand binding, and that mailman is going to give the butler your mail. All right, so the butler opens the door, gets the mail, but the mailman never gets to come in. All right, that butler then takes that mail to another person on the inside of the house, and that is how essentially this works. So the ligand again is the mailman, which it just delivers the message, but he never gets to come into the house, so he doesn't ever get to cross the membrane here. The butler, which is this G protein, is going to take that message to someone else in the house, and it's going to cause a response. So that's all you're seeing here, is just that the message has to go through different channels or people to get something done. So this one's actually pretty cool of how this one actually works, because it actually has to work behind the scenes. It doesn't work directly like these do. So it has to have a few different receptors. A lot of things have to happen in order for the ions to be able to be unlocked through this gate to come through. So with some of these structures that we mentioned at the beginning, the endoplasmic reticulum produces proteins that are released from the cell. So this can be an example. This can be insulin that's secreted from the pancreas. This could be keratin. Essentially, it just produces proteins. So the ER can also be pinched off and make transport vesicles. That's one reason why you always find that the Golgi apparatus is right next to the endoplasmic reticulum or the rough ER. The amount of rough ER is greater in cells producing large amounts of protein for secretion, such as the pancreas that controls your blood sugar. The rough ER also is responsible for producing peroxisomes, which is what produces those hydrogen peroxide, those little vesicles that contain hydrogen peroxide. So smoothie R is going to function. It's going to include synthesis, transport, and storage of different types of liquids or lipids, such as phospholipids and steroids. So that's how your phospholipids are actually made, is part by your smooth ER. So abundant amounts of smooth ER are present within or the testes that make your testosterone. Also, again, it's really abundant in your liver, which is responsible for breaking down your alcohol. Now, unfortunately, our bodies are only capable of breaking down like 2 ounces of alcohol per hour. And I believe that's actually in males. In females, it's actually a little bit smaller. So males are typically better at breaking down alcohol just because we have bigger body masses on average. Not saying that this is always the case, but because we have larger muscle mass or a larger body, usually we're kind of taller, we're kind of bigger, we're able to break down alcohol a little bit quicker versus our female counterparts. But granted, that's not always the case, but on average, typically usually is because we're usually bigger. The Golgi apparatus, again, its main purpose is to modify and package and sort proteins. This is where a lot of our glycoproteins are actually produced. It synthesizes its own molecules, and that's including long polysaccharides, which we referred to in chapter 2 or 3. Lysosomes are small membrane-bound organelles that contain digestive enzymes. They typically have a pH of 5, so which means they're on the acidic side. They're kind of a weak acid. But remember, these are the garbage disposal of the cell, so they have to be able to break down things, just like our stomach acid. They have to be able to break down some of the things that we ingest. So that's one reason why it has a low pH. So here is another clinical view of lysosomes. So there is what's referred to as lysosomal storage disease. This is also inherited from your parents. So essentially, this can be an accumulation of incompletely digested biological molecules within lysosomes. So essentially, lysosomes lose their function in this disease. And if they lose their function, remember, lysosomes are going to break down dead materials and debris within our cells. If it's unable to do that, all that debris and things that was supposed to break down essentially starts collecting. So imagine if the garbage truck it kept collecting, but it couldn't break it down, then it start backing up. Just like a garbage disposal, if you could break down the food to send it down the pipes, it essentially start backing up into your sinks. Well, this is essentially what happens in Tay-Sachs disease. Essentially, affected individuals lack the enzyme needed to break down complex membrane lipids. This is shown in infants that have this disease. They appear normal at first, but they won't start showing symptoms until they are 6 months of age. And typically, unfortunately, by the time that they're 4 years old, usually they essentially pass away. And there's actually no treatment or cure for this disease. So Tay-Sachs is actually a really big deal. So even though that these organelles are very small and minute, they have an important function. And so if everything does not provides its function or its job, essentially it can cause hindrance on a lot of other things. So even though these organelles are small and we can't see them with our naked eye, does not mean that they're not important. Peroxisomes, they're small as well. They can contain actually over 50 different types of enzymes. They are formed initially by pinching from the rough ER. So the rough ER has a lot of different things that it does for the cell. And they can potentially increase in size. And if they increase too big, they actually split into two. So a place where you can see peroxisomes, if you ever ate an apple and you notice that it turned brown, and after leaving it out for a little while, you're actually seeing peroxisomes actually start breaking down that apple, or they're actually reacting with the air and the essentially your saliva when you get bitten into the bacteria that's actually has came in contact with the apple. So peroxisomes are for protection, but it's also for breaking things down, and that uses peroxide. So anytime you eat an apple and you see it turn brown, well, now you know why. It's due to peroxisomes. Remember, cells are made out of or apples are made out of cells, and cells have peroxisomes in them. So now you're understanding why. Peroxisomes also engage in lipid synthesis, so they can actually form oils and lipids. Um, their lipids are formed by peroxisomes, including specialized phospholipids. So this can be present in the heart, the brain, within the liver. So peroxisomes have just more prominence than just breaking things down. It can actually be relied upon by several different organs as well. And of course, the mitochondria, everyone knows that this is the powerhouse of the cell. However, we have to understand that it creates ATP, which is adenosine triphosphate. But we also have to be understanding that it's not just making ATP. It will, even though it is making ATP, but there's a whole process that it goes through, which mitochondria also function apoptosis, which is programmed cell death. So programmed cell death is essentially when essentially a cell has determined that it has lived a long life and it's time to go on. The cell starts going through like a checklist of essentially shutting itself down and breaking itself apart in a proper way. The opposite of that would be necrosis. So necrosis is essentially where all of a sudden that cell is doing just fine, and all of a sudden it explodes. And all the contents inside that cell, all the toxins, all the organelles, just kind of just go everywhere and start breaking down. That's not how cells are supposed to die. Cells are supposed to start degrading themselves properly by breaking down their insides and disposing of them properly. It's just like you moving out of your house. It's like taking stuff, putting in a box, shipping outside the house to the new house or to the trash. That's how programmed cell death is actually supposed to work. Necrosis, which is not programmed cell death, which is abrupt cell death, it's like if someone um, burned down the house or shot at it with a bazooka and blew it, or put a bomb in it and just blew it up. That's the difference. So apoptosis is like moving out of your house into a new house, you're boxing everything up. Necrosis is essentially like someone putting a bomb in the house and just blowing it to smithereens. That's the difference. Um, but mitochondria actually are helping in that. Another thing to think about too, programmed cell death. When we're all in the mother's womb, if you look at your hands, there's this little webbing in your hands. So if you've ever seen duck feet, their their feet have this thin little layer that's in between their toes, essentially. It's called webbing. We actually have that in our hands and feet too when we're developing in our side of our mothers. However, we go through what's called apoptosis, which is programmed cell death, and it actually breaks those apart and degrades those. So now that we have fingers that are nice and separate, but we still have just a tad bit left right there at the very base of our fingers. So that's actually a good representation of apoptosis. And then ribosomes are actually an organelle that are responsible for making our proteins. Um, Anytime you get a vaccination or the flu shot, those vaccines are actually going to the ribosomes, and are making proteins to make antibodies that your body recognizes and will try to fight off or recognize um, your pathogen that's trying to kill you or make you sick. So ribosomes have a very, very, very important function. They're for making proteins. So you can think of it like a copy machine. It's going to take a copy or a recipe, essentially make it and make many copies of it. So ribosomes have a really big function when it comes to making protein. That's how you're made completely of proteins. So ribosomes have a very, very important function. They are for making proteins. Typically, they're going to be around the nucleus or attached to the rough ER. So remember, if they're attached to the rough ER, they're referred to as bound ribosomes. But if they are just floating in the cytosol, they're referred to just free ribosomes. The centrosomes or the centrioles um, are these little spindle fiber looking components or organelles. These are responsible for essentially making up the cytoskeleton, which remember the cytoskeleton, cyto meaning cell, skeleton, just like we have a skeleton, which is going to make up our structure of our bodies. So it has a big role in making those. Also, it has it's another big role is when it is going through mitosis. So if you may remember, mitosis is essentially when cells are getting ready to divide, those chromosomes get pulled apart, and then they make new cells. So centrioles have a really big part in that. So they actually is what is responsible for separating those chromosomes. If they do not separate those chromosomes properly, that's how you end up with Down syndrome. Um, Essentially, you'll have either an extra chromosome or a chromosome didn't get split properly. That's due to these. So these have a role in separating our chromosomes when it's time to split. So proteasomes, which are a major protein digesting organelle located within both the cytosol and the nucleus of cells. So these are also going to help degrade proteins, but they require ATP from the mitochondria to be able to do that. So they're going to essentially break down damaged proteins, proteins that are not folded properly, um, or proteins that are no longer needed by the cell. So they act as a garbage disposal as well. They actually just help the lysozymes as well. But in order for proteasomes to actually work, they have to be targeted. Um, and for those to be targeted, they uses a protein called ubiquitin. So ubiquitin is going to bind to it, and then that is what's going to tell proteasomes to actually start coming and killing it or breaking it down. So these, as well as it says, it works as garbage disposal. So proteasomes and lysosomes both work together to help break down old things that the cell doesn't need or is trying to get rid of. And the cytoskeleton, which we mentioned that it has to do with what gives the cell structure to keep it stable. So you have what's called microfilaments and then microtubules and the intermediate filaments. So microfilaments, micro means very, very small. Intermediate means middle. So you can get it from that prefix of middle or inter. And then microtubules are a lot bigger. Even though they're small, but they're a tube versus a filament. So all these components together, they give structure to the cell. And the thing is, if you don't have structure, you don't have function. So each one of these have a different purpose, but all together collectively, they're for making sure that the cell stays in the same shape and structure. So microfilaments are the smallest. They're included in the plasma membrane extensions and microvilli. So remember, microvilli are going to be in our gut. They're going to help with absorption. Microtubules are the largest component of the cytoskeleton. But usually you're going to see this usually in flagella. and then intermediate filaments. So this is what's going to be really prominent in your skin, hair, and nails. So that protein that makes it kind of hard and dense, that's actually responsible due to intermediate filaments. And then cilia, like I said earlier, or just hair-like projections that extend from the cell surface. And remember, you only have those really in your trachea, in your windpipe, or in your fallopian tubes. So anytime you ever drink water and you get something, you get it going down the wrong pipe and you start coughing, well, your coughing is helping push it up, but also you have these hairs in your throat that are pushing it up too. And so anytime you hear like a smoker's cough or when you're ovulating, it's really pushing that egg to the uterus. Now, interesting enough, if you are a smoker, it actually paralyzes those cilia. So if you're paralyzing those cilia, so when you cough, it's those hairs are supposed to move like this. If you're smoking or an avid smoker, they don't move. They kind of get paralyzed. So it makes it harder to cough up mucus. Also, if you're a smoker, it paralyzes those cilia in your fallopian tubes that are supposed to move like this and push that egg. Instead, it's going to paralyze those hairs, and then you can't move that egg. So it could be potential that you'll have a difficult time getting pregnant if you're a smoker. Also, you can possibly have an ectopic pregnancy, which means that the egg essentially doesn't lodge in the uterus, and it can potentially get stuck in the fallopian tube and potentially rupture and actually potentially kill you. So that's one reason why they try to tell people not to smoke, because it can not only make you have a bad cough, but it could potentially kill you, not only from like the cancer it could produce, but it can also cause difficulties with pregnancy and cause to have a hard pregnancy, to be honest. And then this is actually what microvilli look like underneath the microscope. And so here you see the microfilaments that are giving it structure to kind of to stand up. So they kind of look like a lot of tree roots, but they're microfilaments that are just going to give structure and support. So here you're seeing junctions or membrane junctions. So remember, your cells are kind of placed side by side each other. And to keep them anchored down, they're going to have these junctions or desmosomes that are going to essentially anchor it to themselves and to the basement membrane. So all these cells, they're going to essentially stick to each other like Velcro. So they stick to the neighboring cells with Velcro or desmosomes, as I call them. And then they also use Velcro down here to stick to the bottom so that these cells don't really move or go anywhere. Imagine if you didn't have these cells would just kind of be moving. You'd kind of be like jello, and you'd always be moving. They wouldn't ever stay put. So they have desmosomes that essentially are anchors to each other. So they're anchored side by side, and they're anchored to the floor essentially. And so these create junctions, so it can actually pass materials through these cells, which you see right here. Or they also can what's called a tight junction, where essentially it's like almost like they are sewn together. And that's what you see in all these little kind of these membrane proteins, which is called a tight junction. But essentially, like someone has sewn these cells together so that they are staying attached, so that they don't move or come apart. They essentially create what's called a seal between each cell. So we have the structure of the nucleus, which we understand the nucleus is on the very inside of the cell. They are going, usually you can really identify, you see right here that this whole black spot right here is the nucleus. And actually, well, this is the nucleolus, and this whole thing is the nucleus. So it takes up a lot of space. And so on the inside of the nucleus, we have chromatin, which is just threads. So think about like a whole spool of thread just unraveled inside of here. That is what our DNA looks like. But if you take that thread and start wrapping it up really tightly, it starts to make what's called a chromosome, which we know as this X little structure here. So if you look down here, you see that you look at all these threads wrapped around these histones, also called a octamer, because there's 8 different proteins here that they're wrapped around. But when you start wrapping them around each other and making them coiled or supercoiled, as they're supposed to be called, it makes a chromosome. But if you unravel it, it just makes this really thin string that sits in here. So it actually, our DNA sits in our nucleus. It actually looks like this and not like this chromosome up here. The only time we'll see chromosomes is when the cell's getting ready to divide. That's the only time you'll actually see a chromosome. Most of the time, it's going to look like a thin thread, essentially. So function of the nucleus and ribosome. So it's going to come down to transcription and translation. So transcription is the formation of RNA. So if you remember saying earlier, your DNA never leaves your nucleus. That it's always going to stay there. So when it comes to making a protein, which means we have to go to your DNA and get the recipe to make a protein, because remember, DNA holds it holds the entire recipe for your body. So we have to go in there and get a little bit of piece of this recipe just to make a little bit of this. So let's say your DNA holds all the recipe to your body, but we only want to make a toenail. So we have to flip all these pages to get to this one recipe just to make the toenail, right? But we have to essentially think about the DNA being the DNA being a book inside the library, but you can't take the book out the library. So what I'd have to do, well, let's say the book is also kind of in a different language. So I have to go to the library and I have to write down the recipe in a different language. Then I have to leave the book in the library because I can't ever take it out. And then I go back and then I have to translate it. That is what happens with our DNA. So the DNA never leaves the nucleus. So that DNA is like the book in the library. It never leaves. But I have to go in, read it, and transcribe it. And then I have to leave the library and leave the book in there. And then I have to go back and then translate it. That's what happens in our nucleus. The DNA never actually leaves. And then it's translated into RNA or transcribed as RNA and translated as RNA. And then it has to go to the ribosome where it's going to essentially become a protein. And that's what you're seeing here in this right here. You're seeing transcription happening where it's going into the nucleus and we're making RNA, we're making an RNA copy of the DNA. And then that RNA is going to turn into mRNA, which mRNA, the M stands for messenger, because it goes into the cell and goes out, or goes into the nucleus and goes out of the nucleus. And then it's going to go to the ribosome, where the ribosome is going to make a protein. So that is what actually is happening in transcription and translation. In transcription, we're just transcribing it into RNA because DNA never leaves the cell. And in translating, we're actually making a protein from it. So in cell division, which if you remember mitosis, if you remember the cell phases, which is PMAT, which is prophase, metaphase, anaphase, and telophase, which we can see that right here, which that is actually of the M phase in the whole cell cycle. So at the beginning of the cell cycle, we're going to have growth and preparation, for DNA replication. So before a cell can divide, it essentially has to make more organelles. It has to essentially make everything. It has to duplicate the DNA, which is going to happen in the S phase. And then it goes into G2, which is the same thing, which is just growth and preparation for division of the DNA. And in each one of these stages, it has what's called a checkpoint. So the checkpoint allows it to go to the next phase. So when going from G1 to S phase to G2 phase, each one of these lines is a checkpoint. And so it doesn't allow it to go to the next stage without it making sure that it has met all the certifications for that cycle. So this is kind of like quality control. So when it goes to, well, when it goes from G1 phase to S phase, it has to, before it can make it to S phase, it has to be able to um, certify that everything has happened correctly in G1 before it can go to S. And before it can go from S phase to G2 phase, it has to make sure that everything has happened properly in S phase. If it does not, let's say if it gets to S phase and something goes wrong, it can't go to G2 phase, and actually it stops the whole cell cycle and actually starts, actually gets rid of that cell. It doesn't allow it to go to the next phase. So in order to be able to go to the next phase, it has to pass it. So this is kind of like going from like the fourth grade to fifth grade. You gotta pass fourth grade before you get to fifth grade. But in the M phase, what most people are aware of, it's the M phase, which stands for mitosis. And these are the different phases. So you have prophase, metaphase, anaphase, and telophase. So if we look a little bit more closely, in prophase, we don't really see the nucleus as much, but we see that the chromosomes have wound up, or those chromatin have wound into chromosomes. In metaphase, we see that the chromosomes have lined up on the midline, which is where M phase comes from, or metaphase. All those chromosomes line up on the middle, and then you see these centrioles there right here, which is going to send out fibers that's going to connect to those chromosomes, and then in anaphase, those fibers will pull the chromosomes apart from each other. And in telophase, we can start seeing the two cells getting ready to split, and then cytokinesis is actually when these actually split because it's division of the cytoplasm. So this, you actually got to see some of the organelles still present here during cell division. And so you're able to see some of the centrioles and what they actually take part in. So the centrioles not only provide structure to the cell, but they also help dividing the cell. And you also have what's referred to as interphases, everything before cell division. And so here are some pictures of actual cells dividing underneath the microscope. And so these are nice, pretty pictures, of course, but these are actual cells where you can actually start seeing actual chromosomes actually separating from each other and actually getting ready to divide and to make two cells. And again, here is referring to back to cell death. So all of our cells um, go through this structure of breaking down. However, all of our cells eventually die and they replace themselves. So they replace themselves by going through mitosis. So it's not a bad thing for cells to die as long as they die properly, um, which is called apoptosis. But if they go through necrosis, um, that's a bad thing. It can lead to things like gangrene and infections and things of that nature. But I hope you learned something new. Tune in next time to our series. We'll be talking about cells and tissues. And so hopefully It's my pleasure. I hope you learned something new. See you next time.